Hi, my name is Ida, and I'm a current sophomore in Columbia College studying neuroscience and computer science. I am also a class of 2023 scholar within the Science Research Fellow program. I joined my current lab earlier this year, but over the summer through the SURF program and generously supported in funding by the Columbia College Science Scholars Program, I continued research within the Bartoli Brain Tumor Lab, headed by Dr. Peter Canoll. My project focused upon phenotypically characterizing and testing the potential of novel therapies, specifically ferroptosis-inducing therapies, on our tumor model, which mimics a recurrence of an aggressive glioma, also known as glioblastoma. When it comes to glioblastoma and glioma tumors, it is impossible to think of the brain and tumor as separate entities. Just as glial cells support neuronal functions, they can help sustain a tumor within the brain, which makes glioblastoma especially challenging. In our lab, we primarily focus on that interplay between the brain and tumor microenvironment. As mentioned previously, gliomas arise from glioma cells, so we focus on OPCs, or oligodendrocyte progenitor cells, since they tend to be the main cell of interest when it comes to tumor recurrence information. But the big question that still remains is, how do we go about studying these malignancies? And that is through engineered mouse models. And before I dive into the research side, I want to give some background into the status quo, or current landscape of treatments and therapies available for gliomas and glioblastoma. So to start off, there's usually surgery to primarily remove that highly cellular, fast-dividing tumor core, leaving behind the margin or infiltrative edge. And especially with glioma tumor resection, it's further complicated by the need to preserve eloquent areas, or portions of the cortex that are necessary for sensory processing. And this surgery is usually then followed by chemotherapy with temolizide, and it's most effective at inducing apoptosis in faster dividing cell population. This is, if necessary, it's then followed with radiation therapy, which again also targets that rapidly dividing cell population. I guess now that we think about this, is what is that missing link, and why is it that with this battery of treatments, we still see that glioblastoma has the lowest survival rate of any solid tumor post-treatment? It has the lowest median survival of patients at about only 1 year, and nearly all newly diagnosed treated GBMs always recur. That brings me to the potential cellular basis for these limitations of treatment: fast cycling cells and slow cycling cells. While this is a very broadly speaking classification, it's kind of important for the study to perform. And as the name insinuates, fast cycling cells are the quickly dividing cells, more proliferative, whereas the slow cycling cells are that more quiescent stem-like cell population. That slow dividing cell population is harder to target because it doesn't exhibit that wildly different characteristic that we know of cancers, of growing very quickly. It's rather very similar to glial cells. And what we need to find is where are those recurrences coming from, likely those cells, slow cycling cell populations, what makes them different, and how we can target them. We have all these therapies that are pretty effective in fast dividing cell populations, but what about that large subpopulation of cells that is unaccounted for? In my research, we sought to create a model that mimicked this more slow cycling cell population, which is moderated by an important cell signaling pathway called Notch. Quite excitingly, we were actually able to show that our model exhibited similar phenotypic characteristics to that of a Notch-driven slow cycling tumor, and that these tumors were extremely more sensitive to ferroptosis therapies. Given what we know about the difficulty of treating glioblastoma, the direction this research is heading is very exciting to see. In the future, I really look forward to bringing my interest in neuroscience and computer science to get a better sense of the tumor and larger brain tumor microenvironment interactions. Even despite the challenges this past summer, it was really a rewarding experience, and I look forward to seeing how my research interests will continue to evolve over the next few years at Columbia.